Okay, so as I was mentioning, the dominant and codominant. Now, if you see here, suppose let's say this particular band. So as you can see, this particular band is present here, but not present here or here. So this is basically, if you think about a dominant marker. So in dominant marker, only two things are one thing is taken into consideration, that is whether it is present or not. So this particular band is present in at this particular locus. This is the locus, or this band is present for this particular species, but it is not present in this species. So accordingly, if this is a dominant marker, that means we have to take into consideration either it is present. If it is present, I will write plus. If it is not present, I will write minus, or I will write 1 for presence and 0 for absence. Now, in case of codominant marker, we take expression also along with this presence and absence. So in case of codominant marker, there will be two different aspects. One is either present or absent. Secondly, if it is present, then that differential expression will be taken into consideration, where the expression is very high, where it's high, where it's medium, where it's very low, where it's very low, etc. So depending on how many bands you have, what are the different expressions that you have or intensity that you have found out, based on that you can classify. Like for example, here this particular band, this is present in all these four samples, but as you can see in this one and this one, this is very intense, that is the intensity is very high, and in this one the intensity is relatively low, and in this one this is very low. So these things and so these things are taken into consideration for codominant marker. So either this can be present or absent, like in this samples, this particular band is absent. But even if it is present, then also how much is the expression or intensity, that is also taken into consideration.